Hello, I'm Professor Amy Paller from Northwestern University Feinberg School of Medicine in Chicago, USA, and I'll be speaking with you today about lessons learned from clinical trials. This is an exciting time for EB family and for researchers. We have a new classification for EB that just came out based on new gene discoveries, and we're learning about how the changes in genes translate into the features of EB that we see. There are new treatments moving forward that could someday fix the underlying genetic change or at least prevent the manifestations of EB. We also have the rapid development of new drugs for other diseases, and this can be useful in repurposing them for EB. And as you'll see, growing clinical research about EB, and this is only expanding. So there's a lot of human subject research going on. What is that? Well, it means data collected from humans, and any human subjects research requires informed consent. It can be a survey or an interview. It can be epidemiological research, and that can involve a registry. It can be used for basic science research, either now or in the future through biobanking. We can extract DNA, either from blood, but also from just swabbing the inside of a cheek or having you spit into a tube. We can take tape strips, and that has been tolerated by EB, or do suction blisters and get skin to study without doing a full biopsy. But we can also do a biopsy to look at the tissue under the microscope, to pull out proteins and genes to examine, and to grow all kinds of studies to use as therapy, to grow all kinds of cells to use as therapy or to evaluate. There are also interventional trials. These are ones where we're testing the effectiveness and the safety of a medication or a device or a technique. Now, I would encourage all of you, if possible, to enroll in a registry. There are many. An example is the International Registry based in the Netherlands, which now has almost 700 patients with just dystrophic EB and is characterizing mutations. In North America, we have the EB Clinical Research Consortium that has 20 sites in the US and Canada and almost 1,000 patients in our registry. Another example is the Australasian EB registry with more than 500 patients. And these registries have been helpful for us to study clinical aspects. For example, in our North American EBCRC, we've studied strictures of the esophagus, wound colonization, itch in EB. We've developed best practice guidelines for anemia. We've looked at genetic testing results, and we're evaluating transition to adult care. So why do we do clinical trials? Well, we certainly want to increase our understanding of EB, what causes it, what characterizes it, what problems are associated with EB, how can we most effectively treat it, and how can we treat in the safest way. There are many kinds of clinical trials, small early pilot studies, usually small numbers or even a single center, or multi-center studies, larger numbers of subjects, industry-sponsored, or they could be started by a researcher to ensure validity and reproducibility of results. Are clinical trials safe? They're as safe as we can possibly make them. No clinical trial can be performed without approval by an institutional review board. Drug testing also requires authorization by the FDA or the EMA to do studies in humans. And any new drug that comes on the market has to be tested in this way. Now, you can't have a study done without you signing informed consent. And don't sign unless you understand the risks and benefits. There's always someone there explaining and answering your questions. In many clinical trials, there's what's called the placebo arm. You may be in a group that gets the placebo, that does not get the active that is being tested, but that can be helpful too, as I'll explain. I want to just tell you about some terms. The gold standard of interventional studies is the randomized double-blind placebo-controlled trial. Randomized means that the group you get into is decided at random, usually by a computer. Double-blind, that neither you nor the investigator know what you're getting. And placebo-controlled, that means you might get a placebo. That can be a vehicle if it's topical. It can be a pill or liquid if it's oral. It can be a salt solution if it's an injected drug. But the placebo has to look like the active medicine to maintain that double blind. Now, sometimes you have a single blind because you just can't hide what you're getting to you. An open label means that everybody's getting the real thing. And oftentimes this is a continuation of a trial, or sometimes early in the trial, to see if it works up front or to see if it continues to be working and safe. You have to be aware that there's a placebo effect. And that's why having a placebo arm is so important. Just being in a trial can lead to benefit. It improves your compliance, so you adhere better to the regimen because you're coming into regular visits to a doctor. And because you're being seen by a doctor regularly, you can also tweak your regimen a little bit in other ways. And I think just being in a trial makes you feel better, makes you hopeful, and that also can have an effect on your disease. So because we're looking to see the difference between active versus placebo, it's really important to have that placebo control. That can help us both understanding how effective something is and how safe it is. Now, there are 4 phases in a clinical trial I want to make sure you understand. Phase 1 is very early on, small group, preliminary test of safety, may help to determine the dose, but that is largely determined in phase 2, larger studies, looking at safety and effectiveness, including looking at different drug doses to decide what dose or doses will move forward in the definitive phase 3, which is pivotal for approval by the regulatory body. This is an even larger group to confirm efficacy, to monitor side effects, and sometimes, particularly in Europe, to compare to commonly used treatments. Phase 4 is after it's out on the market. It helps us to look at the drug effect in large populations, to look at the safety with long-term use. Now, how do you find out about clinical trials so you can get involved? Well, one is the DEBRA site. And you can see here just a screenshot of clinical trials that are up on the DEBRA site. You can find them, you can click on them, you can understand more about them. Another site here is in the UK, just talking about ones that are ongoing that are clinical, but also a lot of preclinical trials, those that are done maybe involving cells, maybe involving animals. And here we have a listing of 93 research projects that are cited, and another listing here, 94 in the EB Research Partnership. So almost 100 that are cited right now online, easily available sites. But clinicaltrials.gov is another source I want to make you aware of, because you can see if it's still recruiting, you can find out details about the trials, you can keep track of what's happening and even see results. So here, all you do is you go on, you type in epidermolysis bullosa, and boom, you find out exactly what trials there are. And you can see here that when I do this recently, I see 103 studies on EB. Now, I will tell you that's 9 more than were up there in 2020 and 2 times the number that we found 4 years ago. You can get all kinds of details, including whether you might be eligible. There's also a site showing what trials are registered in Europe. Although I will say that clinicaltrials.gov has a lot of European, even Asian trials that are registered in it. Now, where can you find more information about what's happening with the trial and what the results are? Again, you can see posting of results in clinicaltrials.gov, but an industry will release as soon as it has its data on a press release online. So that can be a good site. You can also see presentations at meetings and publications when it eventually comes out. I hope you've had a chance to glance through this list of a wide variety of different EB studies, because you can find this on clinicaltrials.gov. Not yet recruiting, actively recruiting, no longer recruiting, but not yet with results, not yet completed, but still active, or those that are completed and then completed with trial results. And you can even see one at the bottom here that actually had positive results, very exciting, and is now awaiting FDA approval. Now, here is how you find out if something's been published. Clinicaltrials.gov will tell you about the study, but here PubMed is a great resource open to the public. You can just go on. I show you the URL here. I'm just showing you examples of 2 trials. This is how it looks on clinicaltrials.gov. Here showing you an early phase trial of gene-corrected sheets of skin that are being grafted for recessive dystrophic EB at the top. The bottom one that just came out last year, showing that you can use a long-term available antibiotic, giving it intravenously for patients with junctional EB with a certain type of mutation called a nonsense mutation, and restore laminin 332 and improve wound healing. Now, I want to just show you this example. This was a trial that was a topical product that unfortunately did not reach significance in its primary endpoint, and as a result did not move forward. But because it was a placebo-controlled study, one could look at the results of the vehicle arm and actually learn from it and help us with clinical research. I want to give tribute here to Professor Morrell, who's here, because she was the lead investigator on this study, which showed that just with the vehicle, 7% of the wounds were closed by just 2 weeks and more than 50% by 3 months. Little change overall if you looked at the body in the total wound burden within that 3-month period. So 5% only reduced. But by day 7 and throughout the 3 months, everyone in the trial overall, including the placebo arm, had reduced pain and itch from just being in that trial. A placebo effect perhaps, or maybe it was the vehicle, but regardless, it gave us great information here with what I just told you, plus also that children younger than 2 years of age, wounds less than a month old, total body burden less than 5% of the body surface area were factors associated with faster healing and more wound closure. So this is really important in thinking about criteria for a clinical trial and when your endpoint might be, who you want in the trial or who you don't want in the trial, so you can really see differences versus placebo. And it tells you that if you enroll in a clinical trial, maybe it doesn't lead to a new treatment, but it can give us very important information. Finally, I'm going to talk about a few additional lessons for researchers. One, inclusion and exclusion criteria can make a difference in if the trial is successful. If they're too stringent, you may not get enough recruitment. EB is rare. There are more and more clinical trials out there that are competing. And if we don't have enough people in a clinical trial, you can't finish that clinical trial and you can't know if something is effective or not. There are other issues like so many medications for patients who are with EB, and that may lead to exclusions. And then there are complications that relate to EB, but if they occur for the first time during a trial, they have to be counted as potential side effects of the, even if the investigator deems that they're not related to the drug. If not stringent enough, then you may have a higher placebo rate because you may have patients who are too mild. And also there can be heterogeneity. There can be differences from person to person, even with a certain type of EB that can introduce variability and can threaten results. A paper just came out showing that it is the older patients, it is those with milder forms who are more willing to be part of clinical trials. So think about it, we need more severely affected individuals. Well, what about recruitment? If everybody gets active at the start and then you go into different treatment arms, sometimes that's a way to be attractive, or having a higher ratio of those who get the active drug, 2-to-1, 3-to-1, relative to the placebo can be another way to increase recruitment. Minimizing invasive testing, decreasing the number of blood draws, making biopsy studies optional, especially for pediatric studies, very important. Important to have a placebo arm, but transition to open label really helps with recruitment. Everybody knows they're going to get something at some point. That's not always possible, especially early on, until the safety is known. There just may not be the ability to move on to something until all the data are in and a regulatory body says it's okay to give open label, and then insufficient supply to may, or especially early on, may allow one to have to wait, may require one to have to wait to go into an open label phase. In general, thinking carefully about study design is really important. Strategizing to have the most significant data for the fewest number of patients. So one way to do that is in the same individual. So therefore, to control for variability, having a topical on one side that's active and then the vehicle on the other. Now, there are some issues with that. We have to trust you as patients that you don't switch in the middle of a trial because something looks more effective. But in general, if this is done, it is a great way to look at topical drugs when there are few patients. Choosing the best endpoints, choosing the best scoring tools. So for example, criteria for baseline wound size, we're now starting to get more information about natural history of EB to allow us to know the best wound size at baseline, the best time to end. You wait till 3 months, you're going to get more than 50% who are on a placebo arm for a topical trial. achieving closure. So do it earlier. And then training investigators well. There are very few experts on EB nationally. It's hard to focus on just wounds if you really don't understand EB. Defining the border of wounds can be challenging and easier to see in person than in photographs, that's for sure. So training everyone who's going to be doing these studies and making decisions about endpoints and size, very important. And finally, in the time of COVID, being flexible. This pandemic has really made trials very challenging, particularly for in-person visits, but also for recruiting people. You should know that hospitals, medical centers are the safest place. They require masking. They require people to be as safe as possible. So it's very safe to come in for clinical trials. But up front, thinking about trial design, home visits, patient and parent assessments, flexible timing, keeping visits as short as possible, introducing telemedicine, etc., are really ways to help to recruit. So in conclusion, clinical research advances our knowledge and options for therapy. Click on the Deborah website for learning about clinical trials and also on clinicaltrials.gov. Look for published articles through the Deborah website or other websites as well as PubMed. Study design is critical, but it's shaped through the ability to attract enough participants as well as understanding the natural history without intervention. So my message to any of you who are researchers, get moving on clinical trials. For any of you who are families, get involved. Benefits outweigh the risks if you meet criteria, and look online. Just because there may not be a researcher right next to you or in your town, there's often funding to travel to a site when you have a rare disorder like EB. Do get involved. And with that, I want to thank you for your attention.